Next, we'll look at some curation issues that you might run into with processing your data, either pre-processing it prior to registration or just in general. Sometimes samples have different channel names, for example. So we'll look at this experiment that has just 2 batches. The batches are indicated here by the title experiment name. And we'll give them a reference. And for this reference, since there aren't internal standards, I'm going to specify them as just an aggregate. And for the next piece, we have samples to register. And I want to specify each of these as aggregates as well. So I'm putting a 1 in every sample. So from these 2 batches, the first batch is going to be aggregated and registered to itself. There's an important reason for this. You want to subject all your samples to the same manipulation, just to be fair. And the second batch is going to be aggregated and then registered to this reference as well. And then we want to apply those batch shifts back to the original samples. So I will specify the samples to shift. And again, each one of these gets a 1. And I'll save that and close it. And then we will load that into swiftReg. And we'll select registration settings. We'll leave the mode as batch registration. And for this sample, I think we'll do just neighbor-dependent cluster registration because we don't know exactly what the populations are inside, so we don't want to run the risk of pushing too hard on these samples, especially when they're aggregates. Again, I'm going to, for speed, I'm just going to decrease the concatenate size. I don't recommend doing this if you were doing this on real samples in a real experiment, unless you had a very good reason to use a very small sample size. Normally, we want to keep those numbers fairly large so everything is well represented. And I'm going to pick the file name. For the FCS files, I got to specify a reference. So that was under reference. I need to pick samples to register, so I'll do that. This one, batches were listed under experiment name this time. And then finally, samples to shift. And when we run the validation, it's going to find that there are some samples that have mismatched channel names. So it's asking us, do we want to use Swift modify channels to manually match channel names? And this is desirable, so we want to go ahead with that. And it will launch a different program at this stage to help us with that. And again, all the help for using this program is listed on the right, and it will show you each set of channel names, and you can kind of scan through them and find the differences. And it looks like we're on the left, we have Cy55A, which doesn't appear to be on the right. And on the right side, we have these 2 Q dots at the end. But we do have interferon gamma Cy7A that seems to match between these 2. So the way we would fix this is, I think the most efficient way would be to select these 3 on the bottom and just shift them down by 1. So I'm going to move these channels down. And that looks like it lined up our gammas, and the Q dots are on their own, and Cy55A is also on its own. And so we need to also specify the output channels. And the way we do that is output channels will be populated by anything that you have selected on the left. So I will select the rest of these and then click Set Output Labels. And and that populates our output channels. And it's very important that all channels have a name. And so then we can create new FCS files with the modified channel names. It's going to double check with us. It's going to append the string _MDC for modified channels, and it's going to be appended to each file name before the extension. So it'll be _MDC.fcs, and it tells us that it's going to require about 120 megabytes of space. And we want to continue. It's going to make those new files. And it tells us that it has created a new column in the metadata file that contains those new files. And the next thing it finds is compensation matrices are experiment specific. So the experiments were run on different days. Maybe there were some slightly different machine settings. Compensation, as a result, turns out to be different. This happens quite often. But in this case, I know that we were able to modify the compensation matrix for some of these samples in such a way that we were able to use a single compensation matrix across all of the experiments. It just turns out that that was better in this case. So you have the option of allowing the inconsistent comp matrices, which essentially is allowing for experiment-specific compensation matrices, or you can select to use one compensation matrix across all files, and that's the one that we're going to use. In addition, we have the option of using a CSV file or an FCS file. So if the CSV file is formatted in a standard way, for example, you can get the compensation matrix from an FCS file extracted in FlowJo and saved as a .csv file. Or we can point the program directly at an FCS file, and if it has a compensation matrix inside it, which these do, it will use that compensation matrix on all of the other files. And so that's the option that I'm going to choose. So it'll ask us which one to use, and I'm going to point it at 509322, and it'll use that sample's compensation matrix for all of the other files. And again, once it's done that, it asks us if we want to create the new FCS files, and when it does so, it will append _NCM for new compensation matrix prior to the .fcs, and this will require another 130 megabytes roughly of storage space. So I'm going to go ahead and create those new files. And it informs us once again that it has created a new column in the metadata file so that we can track what we've done. This last question is asked because sometimes you want to do an additional slight adjustment to the data near zero for Cytof files. These are not Cytof files, so we're going to skip the threshold transform, and I'll describe this in more detail in another video. So now it's concatenating the files, and in a moment it will allow us to launch the cluster settings. Now, I discussed a lot of these cluster settings in the batch registration video using internal standards. And one thing that I didn't mention too much was the use of these ArcSinh scaling factors and what they do to the data and why you might want to take a look at those closer and maybe make a few adjustments. But before we do that, first recall that Cy55 and the two QDots were not present in all of the data sets. And so we actually don't want to use these in our clustering analysis. So I'm just turning all of these off. I don't even want the output for those. You can get the output if you want, but we won't need that for any of our purposes. This selection on the far right shows the channels that we can inspect more closely in the histogram plots showing the ArcSinh scaling factors. So again, these are kind of jagged because we're looking at 10,000 cells. But I think a good indication of what the ArcSinh scaling factor does, we can take a look at Avid here, for example. It's defaults to 68. And the defaults that the programs select are optimized. They're not always perfect, but they're very close. And you can often just get away with using whatever defaults are selected. But I'm going to show what happens if the ArcSinh scaling factor is too low with Avid. So I'll select a 1. And what you see is the data separated around the dashed line. The dashed vertical line represents 0 on the x-axis. And the ArcSinh controls the amount of contraction in the near 0 region. And our goal is to have a nice smooth transition for each of these peaks that is in the near 0 region. So if I take this back to 68, we'll see those two separate peaks merge back into one. And this would be a fine setting. If, for example, the ArcSinh scaling factor was too high, what you'd see is the peak would begin to spike near 0. And so, yeah, you'd get this very narrow, sharp peak, and that is not necessarily desirable. This is kind of like the shape that you are aiming for with these ArcSinh scaling factors. And for example, if you had selected a 0, that is telling the program to do a linear transformation on the data, or to treat the data as linear. In the case of these two scatter channels, we often just allow those to stay linear. You can change these to ArcSinh if you like. It doesn't have too much of an effect for the scatter channels, but for the fluorescence channels, it is much preferred to allow the program to select the optimum ArcSinh scaling factor and just go with it. And this is just here so you can inspect the results just to make sure that everything is working as intended. So I'm going to close that and move on with the next step, which is to validate the channel data. When it validates channel data, it's looking for singular values throughout a channel. It looks for correlations, like very high correlations between channels, and it looks for missing values in channels. And if it finds any of those problems, it will let you know and give you some suggestions of how to proceed. And then once that's done its checking, we can begin the registration. Again, it gives us back a list of all the steps that we're going to perform to complete the registration. We have selected a reference sample that is comprised of these 4 samples. It's going to subsample these and concatenate them such that the final concatenate has 10,000 total events. And that will be taken as our single reference sample. In each batch, it's going to take the entire batch and concatenate it into a single batch sample that will also contain 10,000 cells. And that will be done for the second batch as well. And those will each create a batch-specific shift template. And those batch shift templates will then be applied to the individual samples to shift those individual samples in the direction of the reference. And once we've approved that sequence, we can accept the process and it will carry on with the registration. And again, this will take some time. I'm going to fast forward a bit. And the batch registration has completed successfully, so I can go ahead and close this. And we can look at our samples. Yes, they have all been created. And also note that the two interventions that we've performed, these extra pre-processing steps, there's evidence of those working properly. Recall that the samples had modified channel names. That's indicated by the MDC. And then we added new compensation matrices to each of these files so we could kind of harmonize the compensation matrix across all of the different files. And that's why you see MDC and NCM appearing in these file names, and also being carried through to the final step where the _RGST indicates that these files have been registered. And that is registration with a little added curation at the beginning to harmonize compensation matrices and fix channel names.